So you will recall that we talked about the heart being a pump, and essentially it is two parallel pumps in one organ. And when we looked at the systemic circulation, that was the responsibility of the left pump, and it pumped through the aorta and then distributed through branches to all parts of the body to supply cells and tissues with oxygen and nutrients. So those cells and tissues would be delivered oxygen and nutrients from the various branches, and the capillary networks would then pick up the deoxygenated blood and return it to the right side of the heart. So we would have a systemic circulation with capillaries that were responsible for the filtration of oxygen and nutrients into the cells. So if we put our cells in these spaces here, so these would be our cells, and this would depict our systemic circulation. So we can see by looking at this circulation that it was a closed circulation, but we also recognize that because we're delivering oxygen to cells, the returning blood that we've depicted here in veins in the color blue is described as being deoxygenated. So it's delivered some oxygen, not all of it. It's not devoid or empty of oxygen, but it has removed some of the oxygen. So that means that we need a second circulation, and that circulation would leave the right side of the heart, and I'm going to sort of simplify it, and from the right side of the heart would deliver to the lungs, and we'll put the lungs, left lung, right lung, so we simplified that. And then we're going to have our circulation returning to the left side of the heart with fresh oxygenated blood. So the pulmonary circulation and versus the systemic circulation had two different purposes. They each had their own purpose. The pulmonary circulation is about delivering deoxygenated blood to the lungs. The lungs were inflated with fresh air so that they can oxygenate blood vessels, and that's what we're going to learn about today, and return it to the left side of the heart for delivery to the system. Does that make sense? We okay with that so far?
Yeah.
We understand the two purposes of those circulations. So what we need to look at is an airway pathway. And the airway pathway, I'm going to simplify it here. We're going to see some nicer pictures of this in just a minute. But literally what has to happen is we're going to deliver air through the airways to the lungs to meet with the blood vessels that came from the right side of the heart that are low in oxygen. We're going to diffuse some fresh oxygen and bring it back to the left side of the heart for delivery to the entire body. So the point I'm making with our little picture that we've got here so far, even though it's a little bit crowded with some of the wording in there, is that the respiratory system is actually open to the external environment. And that's not a big surprise. Most of us realize that when we breathe, we're going to be breathing air from the environment into the lungs. And when we breathe out, we're going to deliver the air that was in the lungs back out to the environment. And that's the way we're going to get rid of CO2. So we're going to introduce new O2, oxygen, and be able to eliminate CO2 that accumulated inside the body in this way. So this is a very, very quick way to summarize the responsibilities of the respiratory system. So when we look at this, we can appreciate that from a respiratory perspective, there's got to be a pathway for air delivery. There's going to be a location for oxygen exchange. There's going to be a location for CO2 exchange. And that's going to be essentially the same location, but moving in opposite directions. Okay, so these are considerations that we need to be aware of for this respiratory system. And then we also need to consider oxygen delivery to cells and tissues. And we're going to see that this is described as internal respiration because we're down deep inside the body at the level of tissues. And this is the location for what we call external respiration. So when we think about the respiratory system, we want to be aware of the location for external respiration as well as internal respiration. As far as the cells are concerned, internal respiration is about oxygen delivery to the cell and CO2 removal from the cell. So that's what we're talking about when we talk about internal respiration. And as a result, the veins carrying the deoxygenated blood come back to the heart and send up to the lungs in order to replenish that lost oxygen and remove the CO2. So this is sort of a summary picture of the responsibilities of the respiratory system. Now, the other thing to be aware of here is the location of the lungs. We've got two lungs, and most of us are generally aware that they're going to be located in the thoracic cavity. In the thoracic cavity, we would see that the lungs fill up most of the space in this thoracic cavity. And the left lung in particular has a bit of a notch. That's described as the cardiac notch, and the cardiac notch is the space that's made for the heart to sit in. Now, the heart I don't think is going to show up very well in this diagram. Well, maybe it does. So our heart's going to sit in this little space right here. Remember, we said that the pointy end of the heart is kind of attached or partially adheres to the diaphragm below. Now, this thoracic cavity, which is the location for the lungs, is also under what we call a negative pressure. We mentioned this negative pressure when we talked about venous return. But the negative pressure serves a purpose for the respiratory system too, because if it's more negative in these spaces relative to inside the lung, then that's going to help ensure that the lungs are inflated. And from an efficiency point of view, we want the lungs to be inflated under most conditions, because that means that there's going to be air in those spaces that's available for exchange. So remember from our little diagram up above, if I make a bit of space now that we've seen this picture. So we'll draw in a head. Then there's a tongue in there, but we understand that there's a nasal passageway, there's an oral passageway separated by a bit of bone and tissue here, and that represents the pharynx, nasal pharynx, oral pharynx, and then laryngeal pharynx. And the larynx is sort of nicknamed the voice box, and that's part of the trachea. At the very top end of the trachea, there are some cartilage-like features that hold the trachea open. So the airway is held open all the time. And that can be a problem when we ingest food through this same pathway, because the esophagus and the trachea share the same space above the pharynx. And so that means air is going to pass through and can pass into the esophagus, but the esophagus's primary role is to introduce food to the stomach. But because the airway is always open, there's always a potential for food to get into the airway. And most of us have experienced that before. We know what that feels like, right? We start to cough because food is not supposed to get in the open airway. But this airway would continue down between the two lungs and then branch off into the two lungs. So this trachea area would have cartilage rings that hold it open. So if we looked at the trachea over here, we would notice that there are these cartilage rings. Now they don't quite go 100% all the way around. They kind of create a bit of a C shape, the letter C. But on the back, the open end of the C is where the esophagus is located. So the esophagus and the trachea, they'll run parallel with one another through the thoracic cavity, and the esophagus is going to pass through the diaphragm and connect to the stomach. Okay, so that's a separate structure associated with the digestive system. So our air from the external environment is able to pass through either the nose or the mouth and be delivered to the open trachea for delivery through branches throughout the lungs. So the air is going to be taken in through the nose and the mouth and pass through that nasopharynx, the oral pharynx to the larynx, which is the area near the top of the trachea, and enter then the airway special, the unique pathway to the lungs. And this provides us with a pathway for the lungs, for the pathway to the lungs to where the action is going to be. The action has got to happen in a structure that favors air exchange, both oxygen and carbon dioxide exchange. So we'll look at those features as we carry on through this distribution. Now, the other thing to keep in mind, so right on the top of the tracheal opening, we would notice that there are these sort of connective tissue bands, and they can vibrate. Those are what you would call your vocal cords. So as air moves in or out of the vocal cords, sound can be created. So this creates a bit of a top or a cap over the trachea and is where we can generate noise for vocal actions in cooperation with muscles associated with the cheek and the tongue and so on. And that's how we create language and noises that are recognizable for us. And we have to be able to close that during swallowing. So basically, there's a couple of mechanisms that are going to be available to us that make sure food doesn't pass down through the trachea and cause us to choke. So there is a structure that sort of sits above this opening. So it would be up in this area like this. And some of you might know this name. It's called the epiglottis. This opening at the top of the trachea is the glottis. So epi is onto or above the glottis, epiglottis. So we want to be aware of our epiglottis. And what happens when we swallow, and you'll talk a bit more about this when you do the digestive system, muscles associated with a bone called the hyoid, and that doesn't matter for us for right now, are going to pull the trachea up. So the trachea is going to get pulled up towards the epiglottis. It's not that the epiglottis flops down, but rather the trachea gets pulled up to the epiglottis. So the epiglottis forms a bit of a barrier to prevent food from entering. And the glottis itself will be closed in ideal conditions. So in other words, what we should remember not to do when we're swallowing is to talk. So your mom might have told you, or you might have seen this. It's sort of an adage that's discussed in common language. Your mom might have told you this while you were eating. Don't eat while you're chewing, or don't speak while you eat. And really, the gist of that is all about is keeping the glottis closed, the airway closed, while you swallow. So glottis closed and the glottis being drawn up towards the epiglottis is what prevents food from entering the airway. If you decide to talk while you're swallowing, that means by definition the glottis would be open, and you may invite something that shouldn't be introduced to the airway to the airway. The other thing to be aware of while we're on this topic is that our airways are lined by a pseudo stratified and ciliated epithelium. So pseudo-stratified tells us that it is not actually stratified. Pseudo means false or fake. So this is an epithelium that gives the appearance of being stratified when in reality it is a simple epithelium. So if this is our basement membrane that the cells sit on, so the other thing that I should mention in here is that it is a columnar. And if you remember all the way back to week 2 or so of PNA 100, you'll remember that columnar epithelial cells are taller than they are wide. So if we have a simple columnar epithelium, it would look like this. In the pseudostratified columnar epithelium, if we look a little closer, it looks like there's 2 layers of cells. So we've got this layer that's kind of wedged in between that's got this height, and the layer that's got that height. So it gives an appearance of being 2 layers of cells. They're both columnar because they're taller than they are wide. Okay. But the thing that makes it a pseudostratified is that they're both originating from the same basement membrane. So they are it is a total of a simple columnar epithelium. Now, the ciliated part is that these taller cells have these hair-like extensions on them. And these hair-like extensions are able to trap particles from the air, like dust or bugs and different things, or small molecules that might have come from the digestive system. But the idea is these cilia, these hair-like extensions, can trap them, and then they have motility. They have wave-like actions that are designed to send the particles back out. Because as we deliver air deeper into the airway, deeper into the respiratory zones, those zones are designed to be sterile. So we have these mechanisms available to us that are going to prevent things that shouldn't enter into the respiratory zones from entering. So we've got this cilia that have this ability. And you know what sometimes happens is if a little bit larger molecule is trapped by these cilia or possibly escapes those cilia, you're going to cough. If it's lower down in the respiratory system, it's a cough. If it's a little bit higher up in the respiratory system, it could be a sneeze. But in both cases, you're trying to expel something that's entered the respiratory pathway from getting deeper down into where the exchange zones might be. You following me okay with this so far?
Yeah. Yeah.
Okay. Now, the other cell type, that smaller cell type, is a mucus cell. And the mucus is going to be produced by this mucus cell, and it's designed to kind of coat the surface of the airway. So it creates a little bit of a lubricant that's important for the airway, but also another way of trapping particles that might be being introduced. So they might get trapped in the mucus, and then the cilia can wave them back out again. So we've got a couple of examples of protective mechanisms in these airways that come from this pseudo-stratified columnar and ciliated epithelium. Okay, so remember, the goal is to keep the gas exchange locations somewhat sterile, as close to being sterile as possible. And we'll look closer at that location for exchange as we move deeper into the respiratory system. So these airways also serve another purpose. This airway that is the pathway for air down in deeper into the lungs. So the mucus is moist, so that's going to provide some moisture to the potentially dry air. And we'll see that when we inhale or inspire the drier air from the environment, this pathway is going to provide us with a location for moisturizing the air and warming the air, because we can appreciate that body temperature is normally warmer than air temperature. So we're going to warm it up as it passes along, and we're going to try to separate any kind of particles that might be in the air that could interfere with exchange down in the zones where we want exchange to occur. So let's take a look at what our lungs kind of look like. So here we have our air being introduced, okay, through the mouth and nose, and follows the trachea. So these bumpy edges here represent the trachea that has cartilage that helps hold that airway open all the time. So that part of the airway will never close based on healthy cartilage holding that airway open. As we enter deeper down, we're going to do a division into a right and a left primary bronchus. So this was called our trachea. And the first division is a right and left primary bronchus. So bronchi is plural, a single one is a bronchus. Now, what we're going to notice is that from this first division, we're going to divide into additional divisions where the bronchus is going to divide again. And we would call those ones a secondary, but there's two of them. These are secondary bronchi, and they are going to divide into a, in the case of the left side, into an upper and a lower bronchus, because we basically on the left side, we've got two lobes, and on the right side, we're going to see that we have really three lobes. So we've got a secondary bronchus that goes like that and into each of the lobes on each side. So the right side has three lobes, the left side has two, but what you want to really get from what we're showing you here so far is we're getting distribution. First, we got distribution from the pharynx to the right and left lungs, then to the individual lobes of the lungs, and then there will be further branching to get to the actual location where we want to see air exchange to occur. So that's described generally as a bronchial tree. So the bronchial tree looks a little bit like this. Now, I don't know how much you can see. I don't know how big this shows up. At home on your computer, are you able to read the words that are describing the features of this tree, this branching? Can you read trachea? Okay.
Yes.
Yeah, you see that trachea there, left primary bronchus. Then we have our secondaries and tertiaries. And we're going to, but you don't even really have to worry about primary, secondary, or tertiary. Understand that we're going to branch and branch. And as we branch, we're going to distribute to deeper and deeper areas within the lung tissue. And the goal of this system is to distribute air. So these branches are described as conducting branches. In other words, there's no exchange going on in the conducting branches. You can see the cartilage here in these pictures is depicted as a lighter blue color, and they get a little irregular as we get smaller and smaller. They're still present, but they're not in that kind of ring-like structure anymore. They're just going to provide some rigidity, some stiffness to the airways as they get smaller and smaller. And eventually, when we get down to bronchioles, the cartilage is going to be replaced by smooth muscle. And smooth muscle can be targeted by the autonomic nervous system to relax. And if smooth muscle relaxes, what would happen to the diameter of the airway in that bronchiole?
It constricts.
To widen.
You got to speak up.
Maybe it will be widen.
That's right. It's going to widen, which we would call that a dilation. It would be called bronchodilation. So we know, you can appreciate that during fight or flight, we want more air delivery because we want more oxygen to be able to make more ATP. So the messenger for the sympathetic division, fight or flight division, is epinephrine and norepinephrine. So in other words, when norepinephrine and epinephrine targets the bronchioles, we're going to get a bronchodilation. And if the rest and digest branch of the autonomic nervous system, which utilizes acetylcholine as its messenger most of the time, we would get bronchoconstriction, reduced airflow. So we're down here at the bronchioles where we can affect dilation or constriction, and I just use brackets facing each other is a way to sort of illustrate for me and you bronchoconstriction in this case. And when the brackets are facing away from each other, bronchodilation. If you're not happy with those abbreviations, then just write the words out to make it work for you. So the bronchioles will further distribute and end as a terminal bronchiole. A terminal bronchiole is going to end in a cluster of alveoli. And the alveoli well, an alveolus is the site of oxygen and carbon dioxide exchange. So this is where we're trying to get to. This is the functional unit of the respiratory system. And they tend to be arranged in, so here we would have our bronchiole, and it would terminate in a cluster of alveoli. So the terminal bronchiole sort of finishes into a cluster of alveoli. And the branching within that terminal bronchiole is sometimes called a respiratory bronchiole because it does have some ability for air exchange to occur. But this is our unit. Our unit is an alveolus, and we'll have a million or more of these alveoli in each lung. So we started with a lung that looks something like that, and the lung itself is made up of clusters of these alveoli that have to have air distributed to them through the trachea and then the bronchi and then bronchioles, finish in a terminal bronchiole that is surrounded by a cluster of these alveoli. So this is sort of the anatomy of this respiratory pathway. And when we get down at this level, the respiratory bronchiole and the alveolar sacs are where the actual respiration occurs. And that's why we've got this kind of zone differentiated here. And we can distribute the airflow through bronchoconstriction and bronchodilation. When we need more air delivered to the alveoli, bronchodilation. When we need less delivered to the alveoli, we can bronchoconstrict. And that will allow us to distribute air in different parts of the lungs as is necessary. Now, if we go back and look at our picture here, so we can take a look at this picture and appreciate too that from the heart that's located right in this space right in here, remember from the heart, we're delivering from the right side of the heart, we're delivering, oops, I want blue in there, I think. Pulmonary trunk to pulmonary arteries. Remember, these were an example of arteries that carried deoxygenated blood. And this deoxygenated blood is going to end up being branched all around alveoli, where the alveoli can then distribute oxygen and bring the oxygen back to the left side of the heart. And we would see the same thing happening from both sides. Okay, so keep in mind that that's the pulmonary circulation. We had pulmonary arteries, deoxygenated blood that go to the alveoli, pulmonary veins carrying oxygenated blood back to the heart from the alveoli. And again, we're describing our pulmonary circulation here. So this is another way for us to look at a little bit nicer picture of the same idea though. And that is we're looking at our systemic circulation, which works best when we've oxygenated the blood vessels in the lungs, deliver to the left atrium, from the left atrium to the left ventricle, pump through the aorta, distribute through bigger arteries, medium-sized arteries, smaller arteries, to capillaries. Remember, that was about filtration when we talked about the capillary responsibility. Reabsorption to the venous side. Venous return, which was dependent on our skeletal muscle pumps, our one-way valves, and our thoracic pump. The thoracic pump created because of negative pressure in the thoracic cavity. And we get our venous return, our stretch on the ventricular wall, preload, contractility, delivery up to the lungs through pulmonary arteries, back from the lungs in pulmonary veins. This represents our pulmonary circulation. This dynamic of carbon dioxide being able to be removed and oxygen delivered is going to be the responsibility of the conducting airways that enter and leave the alveoli. Right? Everybody okay with that? Right? I've said this now several times, a few different ways, but the same concept is applying here. We okay?
Yeah.
Okay. And down here, you see the arrows that indicate oxygen and carbon dioxide exchange here. We're going to talk more about this as we move along, but that would be our location for internal respiration. So we're going to look at the dynamics of internal respiration when we get down into the physiology. Right now, we're still trying to get our heads around the anatomy. Okay, so this location up here, because of exposure to the air from the atmosphere, from our environment, our external environment, would be our external respiration. Okay, so we've got these zones and these areas we need to be aware of and start to get comfortable with. So when we talk about blood supply to the lungs, you might think, well, the pulmonary arteries must be supplying the lungs. But keep in mind that the pulmonary arteries and the pulmonary veins are part of a pulmonary circulation. It would be the systemic circulation that's responsible for delivering oxygen to the lung tissue. So the lungs are actually going to receive a branch from the arteries on the systemic circulation for delivery of oxygen to the lungs. That's from what we call a bronchiole. So when we talk about the lungs and their blood supply from the pulmonary circulation, they're receiving deoxygenated blood from the pulmonary arteries in order to oxygenate it and deliver it back to the left heart through pulmonary veins. That's pulmonary circulation. The systemic circulation is about delivering oxygenated blood to all the cells and the tissues in the body, including the cells and tissues of the lungs. So the pulmonary circulation doesn't deliver oxygen to the lungs. In fact, it's delivering deoxygenated blood to the lungs for a different purpose, oxygenation or external respiration. We okay with the difference between the two circulations?
Yeah.
Okay. So again, what we want to be aware of with what we've been talking about so far is that we have an airway, a pathway that's delivering air from the external. Let's try to summarize what we've been talking about so far, okay? So let's go here. So over here, we'll draw. Oh, I can't because it's not matched up very well. So we'll put in eyes, nose, mouth. Okay, and then we'll go down into the head and neck. Okay, so we've got our airway. And our air, remember, is going to be high in oxygen and low in CO2. So that's a good thing. So it's going to be delivered through the nose and or the mouth, and from there delivered to the trachea. Remember, the trachea had vocal cords that capped it, and the trachea was open and kept open by cartilage rings until it entered the thoracic cavity and divided into what we call primary bronchi. Primary bronchi would divide into secondary bronchi and then tertiary, blah, blah, blah. And eventually what we did is we created a tree of branches. And these branches that we've illustrated, and we'll keep them in white, are conducting pathways. So that would be the pharynx, the trachea, bronchi, all the way down to terminal bronchi or bronchioles. So I should squeeze in their bronchioles. Not all bronchioles are terminal. Okay, so these ones in white are responsible for delivering air, and they're going to warm the air as it passes through these passageways and distribute the air towards the alveoli. Remember, the alveoli were our functional units in the lung tissue. So we're also going to moisten the air. We're going to warm it up. And that's all part of the responsibilities of these conducting pathways. Then when we get to our respiratory pathways. Those are the respiratory bronchioles and the alveoli. And remember, like I said, the alveoli to the tune of about 1 million per lung. So we got a large area for distribution of the oxygen that we're breathing in and the CO2, carbon dioxide, that we're going to breathe out. Okay, so the next thing we want to just generally be aware of with respect to these zones is what mechanics do we take advantage of for air delivery and CO2, or I should say oxygen delivery and CO2 removal. So that's, in other words, we're talking about the mechanics of breathing. So we'll get to that actually, because I want to finish talking about this alveoli. So when we think about the anatomy of the conducting pathways, we think about those cartilage that get progressively less organized, so that as we move down, instead of being full rings, they may be partial rings. And then as we get into smaller bronchi and bronchioles, they become eventually the cartilage is going to be replaced with smooth muscle, and the smooth muscle can respond to the autonomic nervous system to dilate or constrict, which will help regulate airflow into the segments below those levels, which represent the terminal bronchioles, respiratory bronchioles, and then the alveoli. So the alveoli, let's get this out of here. We're too early on this. I didn't finish talking about the alveoli. So we recognize that additional features to the cartilage are the pseudostratified columnar and ciliated epithelium, and we've talked about what the purpose of that is. Now, when we get to the alveoli is made up of a simple squamous epithelium, which, with what we know about already, we know this is the exchange zone. This is where we want the gases to be able to be delivered to the blood vessels and the CO2 to be removed from the blood vessels. So the shortest possible distance for that to happen is a simple squamous epithelium. So the alveolus is made up of a simple squamous epithelium, and we have the primary cell, the main cell, is going to be what we call a type 1, either alveolar cell, which is a good name, or it's a type 1 pneumocyte. Pneumo meaning air, cyte meaning cell. So we're really saying the same thing two different ways. Just so you're clear on the spelling, because my writing's not the greatest, P-N-E-U-M-O, pneumo. So we have a type 1 alveolar cell. If we call the one cell a type 1, you already know there must also be what? What other cell must there also be out there?
Type 2?
Yeah, we got it. If we identified one as a type 1, it must be because there's going to be a type 2. So our main cell is this alveolar cell, type 1, and it's going to create the simple squamous epithelium of an alveolus. So it's going to provide us with the main appearance. So we're just going to draw in. Here would be our respiratory bronchiole. So it's also going to be simple squamous. If it's respiratory, it means some exchange can happen. So the main cell type is this type 1 alveolar cell. It provides us with our simple squamous epithelium. It will be on a basement membrane, because our epithelial tissues have to have a basement membrane. Remember, epithelial tissues did not have a direct blood supply, so they need to have a blood supply from a nearby tissue. Okay, but at a capillary zone, we've got a simple squamous epithelium here. Okay, and we're going to see later that it is going to be very, very close to a blood supply, which is the pulmonary blood supply. But right now we're looking at the anatomy of the alveolus. So we also need to be aware of a type 2 alveolar cell. Also known, or could be also described as a type 2 pneumocyte. So we're going to see not as many of those. So we might see one sort of wedged in here, maybe one, and another one will just wedge into here. So these would be with the location of the type 2 cells. And the type 2 cells are important cells. They produce something called surfactant. And surfactant is a fluid. And it lines the inside of the alveolus. So these type 2 pneumocytes produce surfactant. So surfactant, we've depicted in this purpley color, is going to line the inside of the alveolus, and technically what it does is reduce surface tension, but importantly what that means is it helps to ensure that the alveolus does not collapse. So this prevents surfactant prevents the collapse of the alveolus, no matter where we are in the respiratory cycle. So you could blow air out as hard as you possibly could, and the surfactant is designed to keep the airway open under all different types of circumstances. So otherwise there would be a tendency for the alveolus to collapse, especially when you think about that negative pressure. If that negative pressure ever became equal instead of negative, where the pressure on the outside is the same as on the inside, it's a possibility that that alveolus would collapse. And that would mean that with a new breath, you'd have to force it back open. So this surfactant helps to improve efficiency. We love efficiencies. So one of the ways that we can improve the efficiency of breathing is to make sure that we produce surfactant. And that's the role for these type 2 alveolar cells. Okay, now we would also see a couple of other cells. There are macrophages, eaters, that patrol the inside of this alveolus. So they're designed to keep the area sterile. If a virus got in there, or bacteria, or even a piece of food, this macrophage would recognize that as being not normally found and would engulf it, would eat it. Endocytosis. Macrophages are big eaters. And we have the odd other type of immune cell that might be found inside this alveolus. But otherwise, this alveolus is a big sac for air. So the oxygen is designed to be delivered into this alveolus so that it is filled with lots of oxygen. Okay, if gas from veins, CO2, is delivered, then the idea with the CO2 is that it's going to be sent in the opposite direction for removal. Okay, so it is this air sac with these two primary gases being part of what keeps that air sac full. Are we okay with the anatomy so far?
Yes.
Let's see, I think I've got a picture I can show you of this alveolus. Let's see what happens here. How about this one? Let's start with this one. So here we can see a cross section of our alveolus. And in this picture here, we can see that the main cell type is going to be this squamous epithelial cell. Our primary or type 1, let's stay consistent with our terminology here. Type, So our epithelial cell is our type 1 cell, and the cell that's going to be producing surfactant is our type 2 cell. So here's our type 2 cell being depicted in this picture here. So it's the one that's going to produce surfactant. The surfactant is this orangey line. There's our macrophage, keeping the environment nice and clean. And we can see a blood vessel nearby. We won't worry too much about that blood vessel just yet. We'll talk about it a little bit later. Okay, now if we want to look at the way things broke down, we'll look at that a little bit later. So in this picture, what we can see is our pathways. So in this picture here, what we're looking at is a section of alveolus with a blood supply. So now we bring it out and look at it a little bit bigger. Here's our individual air sacs. Remember, there's a tune of 1 million or more per lung, and each of those air sacs is going to be perfused or arranged with blood vessels that are going to carry initially the deoxygenated blood. And you can see lots of branching. So if we had an alveolus that I would draw over here, here's our alveolus, and I'll just draw it in blue. Our blood vessels would be distributed all around this alveolus. So what that gives us is a large surface area for exchange. If the oxygen is high inside the alveolus and low inside the blood vessel, then we've created a gradient, a gas gradient for oxygen that favors delivery to the blood vessel. Okay, and that's what this picture over here is trying to show us. CO2 would be moving in the opposite direction. The CO2 would be relatively high in the pulmonary artery and low in the alveolus, so it would transplant the other direction. So if we look at this picture here, let me change my color. Okay, here's our blood vessel with our red blood cells carrying the oxygen. Remember, hemoglobin carried oxygen. Anybody remember how many oxygen molecules a single hemoglobin molecule can carry?
4.
Pardon
2.
me? Pardon?
Is it
2?
4 molecules?
A hemoglobin molecule can carry 4 oxygen molecules.
Yeah.
And in a single red blood cell, there's about 250 million hemoglobin molecules. So for those of you who are quick on your math, that would mean that in a single red blood cell, there would be 1 billion oxygen molecules when hemoglobin is described as being saturated. In other words, it has all 4 binding sites on it available and saturated with oxygen. So hemoglobin was a molecule of a protein molecule that had chains, and it was made up of 4 chains, and within each of the 4 chains, there's a binding site for oxygen. So we have 4 binding sites for oxygen, and then the oxygen is going to bind to all 4. That makes hemoglobin described as saturated. That's what makes your blood look bright red. So when oxygen is delivered from the red blood cell into the alveolus, or vice versa, we're getting an exchange. So when the blood is deoxygenated, hemoglobin is looking for more oxygen, and it's going to get it from the alveolus exchange. But what we're looking at in this picture is we're supposed to recognize that our endothelium is a simple squamous epithelium, and the epithelium of the alveolus is simple squamous, and they actually share a basement membrane. So you have two single-celled layers of tissue, the shortest possible distance in anatomy that you can have for exchange. So you may remember, and you may have to review this, but for simple diffusion to occur, we need a concentration gradient that creates the movement from high to low. Think of that as moving downhill, right? And we want the shortest possible distance, and we want molecules that can easily pass across the membrane. All of those are factors that would favor simple diffusion. So when we breathe in fresh oxygen, we've got lots of oxygen in the alveolus. When we deliver deoxygenated blood to the alveolus from the right side of the heart, it's relatively low in oxygen. So there's our concentration gradient. Simple squamous epithelium matched with another simple squamous epithelium makes the distance as short as is possible. So there's another factor. Gases were lipid soluble, which meant they did not require a channel to cross. They can cross from an area of high concentration to an area of low through the membranes of the cells because they're lipid soluble. So we have all these factors that are making exchange of gases in the lungs favorable. Okay, this picture shows us an alveolus. This whole picture out here is one alveolus and another one right beside it. This represents the blood vessels, how close they are, right? All the branching that we described over there. So lots of surface area for exchange to occur. And if oxygen is high in the air sac and low in the blood vessel, we create movement into the blood vessel. If CO2 is high in the blood vessel and low in the alveolus, we create movement from the high to the low. Do you get that? Everybody okay with me talking this way right now?
Yeah.
Yeah. So again, we're going to build on that. We're going to talk more about it, but you just want to be starting to get comfortable with that at this point. Okay, now what I want to do is talk about one more anatomical feature, and then we can start to look at some of the physiology, some of the mechanics of what goes on. So the final feature I want to mention is that the outside of the lung tissue is covered in a connective tissue that's called the pleura. So the pleura is two layers, just like we talked about when we talked about the epicardium, okay, with fluid in between. So the innermost layer is described as the visceral layer because it's right against the lung tissue. And the outermost layer is called the parietal pleura, and it's got a rougher connective tissue. It's a bit of a protective mechanism, okay? But there isn't a strong connective tissue that protects the lungs. The lungs are considered to be spongy. They're required to be elastic. You can appreciate that they have to be able to expand and return back to their resting position. And that expansion and return is called recoil. And so a lot of the structural component of the lung tissue is going to be made from elastic fibers that give us that ability to recoil. And then the surfactant helps to prevent collapse of the alveoli. So those are two big features that give the lungs their characteristic ability to expand and retract as we change the dynamics of the thoracic cavity. So the fluid in between serves a similar purpose as the fluid did in the two layers of the pericardium, and that reduces friction. The outermost layer is going to help anchor the lungs in the thoracic cavity. The innermost layer is against the lung tissue, which needs to be elastic and able to recoil. And the fluid in between is going to help reduce friction so that that movement can occur. Okay, so that's kind of the anatomical features that you want to be aware of when you think about the lungs and then the actual location for exchange, the alveoli. When we look at the mechanics of breathing, we're looking at the terminology, inspiration for breathing in and expiration for breathing out. Okay, so what's going to happen is that in order for us to encourage inspiration, think of that as mechanics that are going to encourage the direction of air moving into the lungs. We're going to have to change some of the features of the thoracic cavity. Okay, so and then when we want to force air back out, expiration, we're going to change again the characteristics of that thoracic cavity. So let's take a look at our thoracic cavity first. So this is going to be our thoracic cavity. And the thoracic cavity is created by the rib cage laterally and really superiorly, and the diaphragm inferiorly. So we said that typically the thoracic cavity is under negative pressure. The numbers are not important, okay? But at rest, the thoracic cavity is under negative pressure. When we introduce a lung that is receiving air from the external environment, that external environment has got a positive pressure. So what that means is that the positive pressure okay, the positive pressure is going to come from those airways that are receiving the positive pressure air and keeps the lungs inflated. So inside the thoracic cavity, it's negative relative to the lungs, which are then positive. Are we okay with that? Does that make sense?
Yes.
Okay. So if we want to make the inside even more negative, so just think about this for a minute. If we make the inside of the thoracic cavity more negative, we create a bigger differential between inside the lung and outside the lung. And if the outside of the lung gets more negative, it's going to allow the lungs to expand. Does that make sense?
Yeah.
Okay, so we called that inspiration, right? We said inspiration, and we're going to say inspiration requires expansion of lung volume. Okay, so we're simply saying that for inspiration to occur, we want more air delivered. To allow more air delivery, we need more lung volume. So we'll say requires through more air delivery. Does that make sense? Everybody okay with that statement?
Yeah.
If we increase the negative pressure, in the thorax, we can expand the lung volume. So how do we do that? Well, there's a guy out there that created he called it a law. He called it Boyle's law. And so, and that's because his name was Boyle. And what he said was, if you have a given pressure in a space, so in this case, it's our thoracic cavity space. So if we have a pressure in this space, if we increase the space, it's going to result in a decrease in pressure. In other words, pressure and volume have a reverse, or it's called an inverse relationship. If you make more volume, you're going to create less pressure. Okay, and again, we can sort of use the analogy of a balloon. When you blow air into a balloon, you're increasing space, and there will be pressure. But if our balloon's not a good example. I started to think that was going to give us a good example, but that's not going to do it. That's a different dynamic as far as things go. So we're going to go back to our just looking at this space here. So if we say we increase the space, we're going to decrease the pressure, okay? We can increase the space by either increasing the distance, superior to inferior, create it, and also by making it wider and taller. So that would result in a decrease in pressure, right? So are we okay with that? Because you can see we increase the space, and as a result, we're going to decrease the pressure. Okay. If you want to visualize it with particles, if we said we filled up this space with particles of green dots, okay, and when in the smaller space that green is much more dense, okay, as we increase the space, the green dots are less dense, so there's more space for those same volumes to take up. So they're going to be under less pressure. Does that make sense? Am I helping you out here, or making things worse? So if during inspiration, our goal is to make that thoracic cavity space bigger so that we can fill up the lungs more, okay, there's two ways that we're going to do this. We're going to recruit muscles. The external intercostal so those are muscles that are between the ribs. Literally, intercostal tells you it's between the ribs. And what that does is that will pull the ribs up and out. And when we pull the ribs up and out, we increase the volume of the thoracic cavity. And the other thing we're going to do is we're going to contract the diaphragm. And when we contract the diaphragm is going to flatten. So that will increases the superior to inferior distance. If we pull the ribs out by contracting external intercostal muscles, the ribs go up and out. That also increases the anterior to posterior distance. These two things increase volume which means we're going to decrease pressure. So lungs can inflate. If we looked at the rib cage from the side, it would look a bit like this, where the ribs and the spine are formed here. And then when we introduce external intercostal muscles, they're going to expand the anterior posterior distance compared to what it was. So that was what's happening, number one. When we see that in the anterior posterior view, this distance is increased when the diaphragm flattens. So we've increased superior inferior, we've increased anterior posterior distances, which means we've overall increased more volume. We know they have an inverse relationship according to Boyle. So that gave us a relatively increase in pressure with the decrease in pressure. And as a result, the lungs inflate. So that's how we get inspiration. We get this? How did we do it? We recruited and contracted two groups of muscles, the external intercostal muscles and the diaphragm. Okay, so never mind the writing. In inspiration here now, okay, the diaphragm moved down. And the ribs were up and out because of the external intercostal muscles. As a result, volume expanded, lungs expanded, you can see here, because volume went up, but pressure went down in around the lungs while the pressure from the atmosphere stayed the same. As a result, there's a bigger differential and we inspire, we expand. Okay, in the And for the opposite to happen, all we do is stop contracting these muscles. The diaphragm will move back up, and the ribs will return back to their resting position, which increases the pressure and pushes air out, because now the lungs are going to get smaller. And when the lungs get smaller, we push the air out. Expiration. So inspiration, and remember, we're talking about at rest. It's active. We have to contract the external intercostal muscles, and we have to contract the diaphragm. And on average, at rest, remember, at rest is important because this will change if we're getting into situations like fight or flight. Okay, on average, we'll inspire for about 2 seconds, or it occurs, let me finish this. Expiration is passive at rest. So all we do is stop 1 and 2. Those are 1 and 2 up there, contraction. In other words, relax so that the thoracic cavity goes back to resting volume, which is a reduction, and that means that pressure went up. This caused an increase in volume, so pressure went down. Okay, but remember, this is talking about what's going on in the thoracic cavity. The lung expand and the lungs recoil. Lung recoil is dependent on elasticity and surfactant, 2 factors that are important for lung recoil. If lungs are going to be healthy, they have to maintain that elasticity and the production of surfactant. One of the concerns when a baby is premature is that it hasn't yet begun to produce surfactant. So surfactant gets being produced probably somewhere around week 35, 36 of the 40-week gestation period for humans. And if a baby's born much before week 36, say about a month or so earlier, then one of the concerns is it won't be able to breathe on its own very easily because surfactant hasn't started to be produced yet. So they may need to be put on a ventilator to aid with their inspiration and expiration because of this inability to produce surfactant when premature. Now it will be able to start to produce surfactant when as its lungs mature, but in the short term between birth and when those tissues mature properly, the baby may require some assistance in breathing. Otherwise, it's very, very labored and stressful breathing. So when we talk about the mechanics of breathing, this is what we're looking at, the mechanics of breathing. Okay, now one thing to consider is we were talking here about the mechanics of breathing at rest. When we talk about active breathing, we're going to recruit more muscles. Okay, so for example, with inspiration, we had 1 and 2, right? So we're staying with our theme 1 and 2 up there. So let's say for our purposes here, 1 was contracting the external intercostals, 2 was contracting the diaphragm, and additional muscles to increase the thoracic cavity volume. So think about what happens when we're exercising, for example. So we're going to recruit some additional muscles. So muscles that might open up the airways. So we might use muscles to open up the mouth and flare the nostrils a little bit. We might use muscles that extend the spine, because if we extend the spine, we're going to push the rib cage outwards. The muscles of the neck will lift the upper 2 or 3 ribs to help make that volume bigger. So recruiting additional muscles, muscles like the scalenes, they're the ones that help attach to the upper couple of ribs. The extensors of the spine, etc. Anything that's going to help expand. Okay, and so this is active. For expiration, so remember when we were talking about passive breathing or breathing at rest, expiration was just described as being passive. All we did was stop these actions. But if we're trying to breathe at a faster rate, then we're going to also recruit some muscles that are going to help us with expiration as well. So what we're going to see here is we would recruit muscles like, if we had external intercostals up here, we're going to recruit the internal. And the internal intercostals, they're going to pull the ribs down and in. So opposite, whoops. Pull ribs in the opposite direction to what the external intercostals did. And that means that we're going to decrease volume. And we also recruit the rectus abdominis. Now, the rectus abdominis muscle, remember the rectus abdominis muscle is the muscle that runs from the rib cage down to the pelvis, right over the abdominal cavity area. So when we do that, what we do is we increase this. This results in an increase in abdominal pressure, which pushes or forces the diaphragm back up. So that's going to force a smaller volume which is going to increase pressure, which is what we're looking for when we want expiration to occur. So the key thing about inspiration and expiration when it's active is that we've recruited muscles. Okay, so during passive, we maintain a 2 to 3 ratio. So resting respiration is often described as being somewhere between 12 and maybe as many as 16 breaths per minute. If we take the 12 number and we take this ratio and we think of inspiration as being 2 seconds on and expiration as being 3 seconds off, that's a total of 5 seconds for a breath in and a breath out. Don't know if you can hear that. So 2 seconds on, 3 seconds off, over a minute, that's 12 breaths per minute. As soon as we introduce active breathing, we're by definition, that means we're going to breathe faster than 12 breaths per minute. We might breathe as many as 30 times a minute. But the idea is we still make an attempt of maintaining that ratio of 2 on, 3 off, 2 on, 3 off. In other words, a little longer to expire than to inspire. And that's the responsibility of the respiratory regulation centers, which we'll talk about at the end of our discussion on the respiratory system. Okay, so we're going to take a break now, but I want you to be comfortable with these mechanics because that's describing how the lungs expand and recoil that establish breathing in and breathing out. So it's important that we're comfortable with those mechanics. Okay, let's take a break. You'll have a chance to think about it a little bit. And when we come back, we'll carry on with some more information. Is we should be aware of some general volumes for healthy lung tissue. So we can measure lung volume by considering a couple of things. First of all, remember before the break, I mentioned that we have a breathing rate that we consider to be our rate of breathing at rest. And that volume of air that we breathe when we're at rest is described as our tidal volume. I'll write that down in a minute. But that would look something like this. We would breathe in and out, in and out, in and out. And then every so often we might attempt a full inspiration before we come back down to breathing in and breathing out. And then we might see a scenario where we take a breath in again, or out, I should say, and so on. So this volume that we're describing here is about 500 mils per cycle. So we would have a differential here of about 500 mils. And let's write that out over here. And that would be our tidal. So that's the amount of breath that you're breathing. Going to put in another volume under here. This volume would be about 1200 and 1100. We're going to put names to all this stuff. And we're going to come across at about 5800. These are numbers that you do not need to memorize. Okay, so we'll use some terminology that you will be expected to know a little bit about, but I'll make it really clear what you need to know about each one of these as far as that goes. So if we said our tidal volume is 500 mils and the bottom line is 23, then our top line is 28. That's pretty straightforward. So our tidal volume is about 500 mils. Okay, so the other numbers that we want to look at, we want to be able to look at what this number represents, which is the distance running from our 28 to our 58. That's about 3,000. Whoops. And then we need to consider this volume and this volume. So we're going to put some names to some of these as far as that goes. So this would be the third number and a fourth number here. So when we looked at these numbers, we mentioned that number one was our tidal volume. Number two is known as our inspiratory reserve volume. So inspiratory So our inspiratory reserve volume is the extra volume of air that you can inspire over and above your resting typical. So that means that the extra air you could breathe in if you took a really, really completely deep breath, and that would represent about 3,000 milliliters in a typical male. So females are usually about 25% difference. A lot of factors come into play. The overall size of the individual, the fitness of the individual, lots of factors can make adjustments to these. But all I want you to understand at this point is if I was to ask you what the inspiratory reserve volume represented, it's the extra air you can breathe in when you work at it, when you try to breathe extra. So if we said we were breathing at 12 breaths per minute quietly, that's our tidal volume. That's our blue line that's squiggling away down here at about 500 mils in and out. Okay, so our inspiratory reserve meaning there's some extra. That's what number two represents. Okay, and number three is called our expiratory reserve volume. So this is the extra air you could breathe if you forced air out. So this is going to be the volume of air that can still be expired when you force it. So in our diagram here, that represents about 1,100 mils. And then number four, this is an important one. This is one that I would probably ask you a question on. The residual volume. So this is the volume of air in the lungs at any given time. Okay, so this is the air that's still in the, even after the most forceful expiration, there's still going to be some air in the lungs. And this is significant because this is the air that we would still have available to us for exchange, no matter how forceful we were breathing. Okay, so you could breathe out as much as you want. There's still going to be air in the alveoli available for exchange. Does that make sense? Are we okay with that? We understand that? And that represents about 1,200 milliliters. Now, another factor to be aware of is our vital capacity. So the vital capacity, I'll just bring that one up. We're not going to talk about all of them. The vital capacity would be this volume that runs from maximum expiration to maximum inspiration. So from there to there, that represents vital capacity. Now, if we go down here and look at vital capacity, so the vital capacity was our inspiratory reserve volume plus our tidal volume plus our expiratory reserve volume. So if we looked at this, it's number 2 plus number 1 plus number 3. Okay, so we can say it's 1 plus 2 plus 3. And we can appreciate that if we add those numbers, 3,000 plus 500 plus 1,100, we're going to get 4,650. $3,000 plus 500 plus 1,100. Or sorry, 4,600. And the significance of that is that that's the, when you go and get tested for how far you can breathe in and how far you can breathe out, it's going to be this vital capacity. And it will, these are the factors that are going to be dependent on your stature, your position, and also the health of your muscles. So the bigger you are, the bigger these volumes would be. If you're standing or sitting, you're going to be able to influence how much you can breathe in or out. The strength of your respiratory muscles and ability to recoil. In other words, the elasticity of the tissues in combination with that surfactant that we talked about earlier. So these are all factors that would play a role in how good you can breathe in and breathe out. And as a result, you create what's called this vital capacity. Okay, so there's some other volume terminology that you could look at if you want to in the textbook or the PowerPoints, but these are the ones to be aware of generally, not to be aware of the total numbers. I'm not going to make you memorize numbers, but just the definitions of what each of these mean. And then in the case of expiratory reserve volume, the significance of that is it's the air remaining in the lungs no matter what phase of the cycle for breathing you might be in. And that means there's always air available in the alveoli for exchange to occur. So when we talk about breathing, remember we said right from the very beginning, we've always said that it's important that we recognize that the purpose of what's going on here, the purpose of the respiratory system is gas exchange. We want to deliver oxygen to the circulatory system and remove carbon dioxide from it. So there are some factors that we need to be aware of that make this better under certain circumstances. So generally speaking, there's 3 factors that make respiration efficient. And these factors are going to change based on things like aging or circumstances, or even the environment you happen to be located in. For example, if you go up into altitude, the amount of available oxygen is reduced. So that's going to play a major factor on how effective respiration is going to be and require adjustments. Okay, so one of the factors that we've touched on, but we haven't spent very much time talking about yet, is the partial pressures, what's called a partial pressure partial pressures of our gases in solution. So when we say in solution, we're, for example, within air, we've got contributions from oxygen and carbon dioxide and nitrogen. So those are all representing partial pressures. And the idea during respiration is that there's going to be gradients where oxygen is higher on one side of a membrane than on another, and that's going to favor movement. And CO2 will be higher on one side or the other, and will favor movement under those circumstances. And what we're talking about with respect to the gradients is that the movements will go in the direction of high to low. So when we look at breathing, both external and internal respiration, we're going to be looking at those partial pressure gradients and which direction that movement is likely to occur. So we'll be talking about partial pressure gradients in just a minute. Another factor is what we call ventilation perfusion matching. Ventilation is about the airways. And perfusion is about the blood flow. So we're talking about ventilation of the alveoli versus perfusion by the pulmonary and then the third factor that we've mentioned, but we're going to emphasize it here, are the structural characteristics of the respiratory membranes. So we've already talked about those briefly, but we'll just mention them again. So in this example, what we're talking about is, let's, if we look at our simple squamous epithelium, and we'll keep things nice and simple. Remember, these were our type 1 alveoli or type 1 pneumocyte. And our respiratory bronchiole. And we might have a couple of type 2 cells interspersed that are responsible for producing the surfactant. So we've got our surfactant that will contribute as an inner lining. Okay, and then we had our capillary. And our capillary was a simple squamous endothelium. So we'll just draw our capillary this way. And because we're up at the site of the alveolus, what we're discussing here would be considered to be external respiration, because the air would be being delivered to the alveolus through or from the external environment, from the air that you breathe in. So we know that it's going to be high in O2 and low in CO2. Okay, and this is our blood vessel, and it would share a common basement membrane. So the two simple squamous epithelia are essentially sharing the same basement membrane. So when we talk about the characteristics of the respiratory membranes, we're talking about the linings of the alveolus, simple squamous, and the linings of the pulmonary capillaries, simple squamous, with a common basement membrane between the two. So again, I mentioned this earlier, shortest possible distance you can have structurally between a one zone, our alveolus, and the other zone, our capillary. Now, in the capillary that's coming back to the heart and being pumped to the lungs, we called that a pulmonary artery that delivered deoxygenated blood. Is that everybody's okay with that, right?
Yeah.
So our deoxygenated blood is relatively high in CO2 and relatively low in oxygen. Everybody's okay with that?
Yep.
Okay, so the idea is that as this capillary network flows through the alveoli of the lungs, this concentration gradient, this partial pressure gradient, favors oxygen being delivered to the capillary and favors CO2 being delivered to the alveolus. Does that make sense?
Can you repeat it, please?
Well, let's take a look at it. This is a visual now. We got lots of oxygen in the alveolus and relatively small amount of oxygen in the pulmonary capillary. That favors diffusion down the gradient. Partial pressure gradient from high to low. Why? Because gases are lipid soluble. They do not need a channel. They can pass through a cell membrane. Shortest possible distance. Two flat cells with one common basement membrane between them. That's the structural characteristics. This is setting us up for simple diffusion from high to low. And if you look at the carbon dioxide in blue, we're going from high to low. Do you see that?
Yeah.
Okay, so what about the person who wanted me to repeat? Do you see that? The
Hello?
person who asked me to repeat
Yeah,
that.
I can see, but I'm kind of lost, but go on.
Okay, all
But
right.
it's,
Well, we're gonna build on this. We're
yeah.
gonna keep building on this. I'm gonna show you another picture and I'm gonna draw this on more than one way. But
Okay.
the idea is, first of all, do you see where we get the high concentration of oxygen? Because we just breathe it in,
Yes.
okay? So you understand where the high oxygen is. Now, the low oxygen is because we're returning from the right side of the heart, deoxygenated, less oxygen blood, because we delivered it to the cells earlier. So that means that the blood going back to the lungs that was pumped from the right side of the heart is lower in oxygen.
Okay.
So that created our gradient.
If
And
I,
the reverse, right?
yeah, can I say
And the
something?
reverse is true for carbon dioxide.
Let's see if I can understand what he's saying. The pulmonary capillaries is where the exchange takes place, or the alveoli.
Is it not true that before?
Is it? Hello?
Yeah, I'm here. I can hear you.
You can hear me, right? Okay.
Yeah.
I just want to understand something. When we breathe in air, into the lungs,
Yep.
where the blood exchange takes place, where on the left side of the lungs, where you have the exchange between the oxygen and carbon dioxide, which is where the oxygenated blood goes back into the system, right? So
Okay, so
this, if
it's
the,
not the left side of the lungs. Nothing about the left side of the lungs.
okay,
It's going to be the entire lungs.
the
Everything up in
entire
the
lung,
lungs
okay, the entire
is
lung.
right and left.
Okay, so when this oxygenated blood that is highly enriched with oxygen goes back into the system, it goes, like the oxygen now goes through it goes to the alveoli, right? Where is the alveoli the organ where the exchange takes place, or the pulmonary capillaries?
So in the pulmonary circulation, the exchange occurs in the pulmonary capillaries, correct?
Okay, so if I can understand you, that when it gets to that, when it gets into the alveoli is enriched with,
Okay, sorry. When it gets into the alveoli, yes?
yeah, the alveoli is enriched with oxygen. That is a higher concentration of oxygen, and then it has a lower concentration of CO2, which is carbon dioxide, if I can
Right.
understand. Then when it gets to the exchange site, which is the pulmonary capillaries,
Yep.
what goes up, that's what goes out of the pulmonary capillaries as exchange, which is the oxygen, is lower in concentration. Why? The CO2, which is the carbon dioxide, is higher in concentration. Is that what you're saying?
That's exactly what I'm saying. Yep.
Okay, go on. Thank you.
Okay. So, and this little, let's try and make it, okay, so let's do this. We're going to look at internal respiration while we're on this topic. Okay, so over here, we will look at what's happening at internal respiration. So external respiration is happening at the lungs, and internal respiration is happening down at the cells. So this is pulmonary, and this is systemic. So we're going to draw in our example of a cell. Okay, this is a metabolically active cell. There's the nucleus. And when a cell is metabolically active, it's using oxygen to make ATP. So we can appreciate that oh man, my pointer is offset and it throws me off like crazy. Because I can't line it up. So if I'm more in the middle of the screen, I can make it work better. So here's our cell. There's our nucleus. And our cell is using up oxygen to make ATP. So it's going to be low in oxygen. And when we make lots of ATP, we create lots of CO2. Okay, everybody okay with that?
Yes.
Okay, so now here's our blood vessel.
Very simple.
That is carrying arterial blood, oxygenated blood to the cells of the body. Okay, so it's going to travel in this direction. Up here, we were traveling in that direction. That doesn't matter so much at this point. But our oxygenated blood is high in oxygen and low in CO2. Right? Everybody okay? So this is coming from the heart. Left side. This is also coming from the heart, but right side. Okay, we okay with the anatomy so far? Yes, no?
So I just have a quick question. So
Yeah.
for external respiration, it's deoxygenated blood, and for internal respiration, it's oxygenated blood.
Yeah.
Okay, thank you.
So we're going to move from high to low, from high to low. Down at the tissues, the result of that is that the cells get new oxygen and get rid of the CO2. Up at the lungs, we deliver the CO2 to the alveolus to breathe it out. We breathe in new oxygen, create a high oxygen environment for delivery to the blood vessels from the pulmonary circulation. So we can make this look. Now I'm winging this one, so we'll see if this works. So let's make a single alveolus, okay? We'll make this as big as we can so that we can follow the volumes and the concentrations and all that. So here's our simple squamous epithelium that's going to represent our alveolus. Okay, here's our blood vessel, and we'll actually make it carrying deoxygenated blood to the alveolus. From the right side of the heart. Okay, so we returned okay, we'll come back to that. So what's happening in our pulmonary circulation is that our left heart and our right heart have two distinct responsibilities. For the pulmonary circulation, we delivered blood from the right ventricle to the lungs, right? And our blue blood is low in oxygen, but it's high in CO2. And our alveolus is low in CO2 and high in O2 because it got the oxygen from the air. So our air came this way and our blood came this way. We okay?
Yes.
Okay, so high concentration, low concentration favors downhill, so that when we do that 1,000,000 times in one lung and 1,000,000 times in the other lung, we're going to get very effective diffusion, so that the arterial blood now that's going back to the heart is high in oxygen, coming through the pulmonary vein, but going to the left heart. It's going to be very high in oxygen. And look at where the CO2 went. The CO2 left the blood because it's going from high concentration to low concentration. And then when we breathe out, we're going to expel the CO2. We're trying to bring in the O2, right? So this represents external respiration. We good?
Yes.
Okay. So now we're going to bring the nice fresh blood back to the left side of the heart. And from the left pump, we're going to leave and deliver to all the cells and tissues in the body. Right? So down here, we have blood that is high in oxygen and low in CO2. Remember, the situation in the cell was high CO2. Why was the CO2 levels higher in the busy cell?
Because you're using it for
The
ATP.
working-
You're
yeah.
using it for ATP.
Right. It's a byproduct of making lots of ATP. We're going to produce CO2. And because we're making lots of ATP, we're using up oxygen. So in the cell, the oxygen levels are low. So what does that mean for exchange? High to low. So we're going to deliver oxygen in, and we're going to remove the CO2 because it went from high to low. So now on the venous side of our systemic circulation, we're relatively high in CO2 and relatively low
In oxygen.
in O2. And we bring it back to the right heart. And when we pump it to the lungs, look at what we have. Low O2, high CO2. We just connected everything. Where is it going to change? It's going to exchange. It's going to change at the external site or at the internal.
Internal. Okay. Those are the two places exchange takes place.
Makes sense.
Yeah.
Yeah.
Okay.